design organized by NIELIT Guwahati. Now, we are going to proceed with the first technical session of the day to be delivered by Dr. Saurov Mahanta. The topic of the session is Introduction to the Tools and Prerequisites for Computer-Aided Drug Discovery. Before moving further, I'd like to take the privilege of introducing Dr. Mahanta. Dr. Saurov Mahanta is currently serving the National Institute of Electronics and Information Technology, NIELIT Guwahati, under Ministry of Electronics and Information Technology. His area of research is bioinformatics-based drug discovery, including medical biotechnology. Dr. Mahanta did his master's in molecular biology and biotechnology from Tejpur University, Tejpur, Assam, and he received his PhD degree from Guwahati University, Assam, for his work on computational drug discovery for tuberculosis. Dr. Mahanta has been actively involved in bioinformatics teaching at the postgraduate level since the year 2003 in topics like drug discovery research using combinatorial chemistry, molecular docking, molecular modeling, molecular dynamics, and related analysis. He has to his name several international publications on structure-based drug discovery studies on COVID-19, breast and stomach cancer, Alzheimer's disease, and tuberculosis. He is also involved in nanoinformatics-based research using proteins of different pathogens. With this brief introduction, I now request Dr. Saurov Mahanta to present his lecture.
So thank you for introducing me, although I was speaking from the very beginning in the morning. So once again, I welcome all the participants to this day-long webinar hosted by National Institute of Electronics and Information Technology to discuss various aspects of computer-aided drug discovery. So my topic, it was assigned to me, is basically I have to talk on the tools and prerequisites for computer-aided drug discovery. Okay. Well, prerequisites for many of us. Okay, in a lighter note, so T is very essential, including me, because means we start the day, we start some work after a good cup of tea, right? So why I am mentioning this tea? Because when we talk about tea, okay, we are not only talking about a very refreshing drink, but also we talk about some of the medicinal compounds that we consume along with the cup of tea, right? So I'll be little bit of kind of informal while delivering the presentation, because means these are some of the topics, means computer-aided drug discovery, molecular dynamics, talking, molecular modeling. These are some big words and very new to many. It may be very new to many of you, and many of you are very much familiar because many senior professors, many senior researchers are there in the group. So I don't know whether I am the right person to speak or not, but I'll try to share some of the experiences that we had in the last 17 years after joining National Institute of Electronics and Information Technology. And we did several work where we got the challenges to start our work. Those things we will discuss, and some way or other you will find the prerequisites in between. Okay, I will not directly go to the specification of the computer, specification of the server, that all is available in the internet. You can search, okay? But the first and foremost prerequisite is the need for a drug, okay? So why we need new drugs? Drugs are available in the pharmacy. You will go, you will search for drugs. You will get lot of different drugs available for different diseases. Except yes, some of the new diseases like the COVID-19 and cancers, some kind of cancers, and some of the diseases which are not curable with the drugs at this moment. But yes, certainly we can say that there is a need of some newer antibiotic molecules, newer antimicrobial agents to fight with different infectious diseases, particularly in the developing countries. Okay. Oh, again, I am putting one image of a very delicious recipe of India, that is khichdi, right? Dal khichdi. So it is loaded with nutrition, protein, but at the same time, again, we are consuming some of the medicinal compounds along with our lunch, dinner. Okay, we take a lot of herbs, a lot of vitamins. Okay, then in between, as Indian, we use lot of spices which are very much healthy, very good for keeping ourselves healthy. They also have a lot of different kinds of medicinal properties, medicinal compounds. Okay, so as the group I can see is there from different part of India and countries like Bolivia, Poland, Taiwan, Thailand, Pakistan, and Bangladesh. Okay, available in this group. So it is very interesting to see the spices, Indian spices, or the spices as some medicinal plants. Okay, tea, spices like turmeric, ginger, pepper, big cardamom, green cardamom. So these all are having different types of medicinal compounds in it. Okay. Then in between the lunch and the brunch or the breakfast, we take some healthy drinks. Okay, like one to mention is amla juice, or say you take some fruit juice, loaded with lots of chemicals. Sorry, phytochemicals, of course, and lot of vitamins like vitamin C, vitamin K, and along with that, fortified with lot of compounds which are very much necessary for our for keeping us ourselves healthy against many diseases. So the first question what uh, comes to our mind, we need new drugs. So why we need more drugs or new drugs? Because our existing drugs are getting resistance over different microbial species like tuberculosis. So you will be surprised that the tuberculosis is the leading cause of death in the developing countries for the HIV patients. People doesn't die from HIV directly, but opportunistic infections, mostly by tuberculosis, comes as the killing agent for the HIV patient. So that is because we have multi-drug resistant variety, we have totally drug resistant strains, we have extensively drug resistant strains available in the environment. So from where those comes? They come basically due to the uncontrolled use of the antibiotics, due to poverty, due to lack of knowledge, or due to many other reasons. We generally do not complete the antibiotic courses, what is being prescribed by our respected doctors. So that way, we give the chance to the microbe to develop resistance against the antibiotics. So to fight with those diseases, those disease-causing agents, we need more new molecules into the clinic. So from where we get those molecules? Chemical synthesis, yes, one answer, but I prefer it from the nature, because all the natural agents, disease-causing agents, I believe that we have, or nature has, some kind of solution for that. And from the natural sources, again, I prefer to use the plant sources. That means keeping aside the technological needs for computer-aided drug discovery, first of all, we need the sources from where we will get the new compounds, right? So that, for today's discussion, I am keeping it as from the plants. That may be some other sources also, but here, to limit the discussion with the plants, I'll start the identification of the plant product. So how we will go ahead with that? So we use it as a food, as medicine, as nutraceuticals, as functional foods, as intoxicating agents, right? So there are a lot of plant-based products available, and right from the morning to the evening to the night, to the bedtime, we use different types of plant products. Okay, so how we can utilize those plant resources to get the answers against the deadly pathogens? So that is one of the major aims and objectives of the computer-aided drug discovery, and computer-aided drug discovery can help tremendously to identify new molecules to be forwarded as a lead molecule for development of a drug. So two very commonly available plant displayed in the screen. One is Tulsi, another one is Neem. Okay, so they are basically the gift of nature. Apart from giving the oxygen, they are giving us a lot of other medicinal plants, medicinal compounds, active medicinal compounds. So we generally believe, you also agree, that when there is some kind of ailments, disease like cold, cough, and even in case of this COVID-19 situation also, we are looking towards our grandparents. We are looking towards our older generations. What they used to do that when there was a situation like that. Okay, they definitely had some of the very good answers to that questions, to our questions. So you take this, you take that, and in many cases it works. In many cases it doesn't. Right, but those traditionally used medicinal plants, if we do some organized scientific analysis, we can definitely will be able to identify some of the active compounds, specific active compounds available in the plant as a secondary metabolite or some phytochemical. So we can have some better understanding of their active principles, active compounds, and we may not get the adequate amount of the active compound while having the tea in the way what we are having. So we can there is a chance of washing away of the unwanted compounds and zooming down to the wanted or the active compounds. So biochemical studies, we can do the chromatographic analysis, like high performance liquid chromatography can be used, HPTLC can be used. Say gas chromatography and mass spectroscopy, or liquid chromatography, mass spectroscopy, or any other chromatography, mass spectroscopy can be combined together to detect the phytochemicals available in the plants. Those software which are associated with the GCMS, LCMS packages can give you a better understanding of the active compounds present in a plant species, present in a sample what you give for GCMS, LCMS, or any kind of chromatographic and mass spectroscopic analysis. And if you zoom down into those molecules, there is a technique available like NMR or X-ray crystallographic techniques for the small molecules. You can go even to the level of the structure determination of those compounds, whether they are actually structure determination in their actual conformations and all. Okay. So GCMS data, what we get in the form of peaks. Peaks can be detected through the GCMS associated software. Many of you are familiar with to determine the volatile compounds, and LCMS is used to detect the non-volatile compound. Combination of GCMS, yes, it can work wonder. So it is not very difficult to do also. There are many laboratories in India or in many advanced countries that is having the facilities of GCMS, LCMS. We can determine the compounds from the peaks, and after getting the compounds, we can study those compounds against the different disease causing agents. Through what? First, through the computer aided drug discovery. So the first prerequisite, as I have mentioned, is we have to have a indigenous, we have to have a own database of compounds. Otherwise, we can depend on the databases like Dr. Duke's phytochemical database, PubChem compound database to get the compounds identified against the plant species, a particular selected plant species. But please don't expect that all the plant species will be available in that those databases. So we can use the analytical, biochemical analysis. We can do the analysis, and we can have the compounds detected. And we can, with the help of the computational techniques, we can identify or we can prepare the combinatorial libraries of those plant compounds. From one compound, we can generate thousands of millions of compounds with a similar scaffold and all. Then we can test it against some of the diseases where you want to test it. So in this short presentation, I'll take the example of Alzheimer's disease and COVID-19. One is infectious, another one is non-infectious. Okay. So in the Alzheimer's disease, there is the huge literature you will get in the web. Huge literature is available describing the AD pathogenesis, Alzheimer's disease pathogenesis. So from there, we can selectively use some of the pathways through the pathway databases like KEGG pathway database, BioSeq database collection, MetaSeq metabolic pathway database. These are three examples I have given, but there are many. KEGG pathway database is the most popular one, which can be used to identify some of the specific metabolic pathways where we can target to design or to target to get some compounds identified from the natural sources to block those metabolic pathways or signaling pathways, okay, reactions. But where we will target definitely into the enzymes which are involved in those metabolic pathway. So identification of the disease targets that will be dealt by Dr. Setia in the next presentation. So how to identify, but just I'm giving one glimpse that what are the prerequisites. So we need the disease, targeted disease, which target disease we will target. So in the disease, we will identify the disease causing organism. From the literature, we can find out the targets, what are the drug targets popularly being used in those diseases. Then, say for example, in case of the Alzheimer's disease, we can specifically include the enzymes which are helping the progression of the disease pathogenesis, and also some of the extra genetics, epigenetics modifications associated with the Alzheimer's disease can be targeted. Epigenetic targets like HDACs, so they are very interesting targets for cancer and Alzheimer's disease. So HDACs are can be used as a target for Alzheimer's disease. So where we can try to identify compounds from the plants, from the active compounds of the plants that is being traditionally used against the neurodegenerative diseases, neurological disorders by our grandparents, by our, say, in the different tribes. Okay, so that knowledge we can gather, we can do GCMS, LCMS, the compounds. So this is about the Alzheimer's drug development. What the situation, current situation. A lot of research is going on in Alzheimer's. So why it is I have selected, because it is not related to the pathogenic intervention. So this is a disease which is basically neurodegenerative disease associated, and there are many factors, epigenetic factors and genetic factors for causing the Alzheimer's disease. So these kind of diseases also can be targeted. Then in the next step, what we will see after identifying the target, so we'll go to the databases like protein data bank or structural databases to find out whether the identified enzyme or identified protein which you have identified as a drug target or the receptor has the structure or not. Structure means the three-dimensional X-ray crystallographic or NMR developed structure. Okay, so if it is there in the protein data bank, we can download the structure of those receptors and we can use it in our CADD study. So different plants, say traditional knowledge base plants we have selected. So for Alzheimer's disease, identification of the compounds, then identification of the Alzheimer's disease targets, then we take structure of those proteins downloaded from the protein data bank. Okay, one thing, if the target structure is not available in the protein data bank, what we can do? Okay, so what we can do is there is also a solution called homology modeling in bioinformatics. If we have the sequence of the protein or identify a drug target, then we can do the homology modeling using very recent homology modeling methods. Like one of the software is very popular nowadays, that is called AlphaFold. This is artificial intelligence based homology modeling software. So very efficiently we can model the near natural structure for a protein. Then small molecule databases, as already mentioned, we can, if it is not available, facility for doing the GCMS, LCMS is not available with us, we can go to these kind of databases and we can get at least some of the compounds for that plant if it is available there. Now, in the infectious disease category, I have kept a viral disease that is creating a pandemic across the world, and why we are today meeting virtually. So it made a lot of changes, the way what we do the research, way what we share our knowledge. Earlier, webinars and other things were not very popular as such, but nowadays it is a part and parcel of our learning life. So you know that it is a pneumonia of unknown etiology. which was first reported in Wuhan city of China, and it is creating a lot of havoc in our country also. In India, develop vaccines. Vaccination is going on in full swing, but antiviral drugs. So scientists are struggling for antiviral drugs. So DRDO has recently means identified one drug. There are several antibiotics which have been used against the SARS-CoV-2 on the basis of the computer-aided drug discovery work. How? That just let me explain. The screening of already available drugs, the first work what we did or the researchers did across the world, basically after the structures of those important proteins, as Borasar's presentation, you have noticed that he has mentioned about the main protease or papain-like protease. So there are some important protein. There are 29 proteins available in the SARS-CoV-2 genome proteome. So out of that, many are designated as good drug targets. So against those drug targets, the scientist, the computational scientist, using artificial intelligence technique, structure-based drug discovery technique, the repurposing exercise. Repurposing means the use of a already available FDA approved drug against the SARS-CoV-2, and up to many extent they became successful also. So those repurposed means already available drug structures in the databases you will get in the drug bank or FDA approved antibiotic list. From there we can track the structures of the small molecule, and on one hand we have the structure of the large molecules of the proteins of that virus. Then computationally we can dock. For the docking study we can identify that which molecule will better fit in a drug target or identified drug target. Then discover new medicines or new compounds from different sources like microbes, plants, etc., etc. That is another line. So in the other line, so we have put our efforts to screen the compounds from black tea, to screen the compounds from green tea, to screen the compounds of known antiviral plants like neem, Houtonia cordata, okay, and we have identified quite a few compounds which are helpful for binding into the designated drug targets like proteases, or say that non-structural protein, other non-structural protein groups, or the structural protein groups, that we have reported many compounds. Not many, a few compounds which requires further study in terms of the cell culture, enzyme assay, and finally we can recommend that yes, this compound can be used as an antiviral or anti-tuberculosis or that that agent, that pathogen compound identified from this plant. So what is the advantage of a plant origin compound? So it has generally, if you follow the rules of drug discovery like Lipinski rule, host rule, these are nothing but some of the parameters of the small molecules identified which are put it together to the Lipinski rule of 5 or say Ghost rule or Weber's rule, etc., etc. They have certain criteria that one of the criteria of Lipinski rule is the molecular weight should not exceed 500 AMU or vice versa. There are so many other uh, means parameters. So on the basis of those parameters, we can filter out those unwanted compounds of those plant, and we can use only the compounds which have the potentiality to become a drug. So we can identify the base binding ligands, base binding compounds, and we can put forward other techniques of say QSAR, admit-tox studies, etc., etc., to do the more validation of those compounds. So this is the drug discovery new compound product development scenario in SARS-CoV-2. These are the mainly used compounds, targets, those red colored marked in red color, these are proteins of SARS-CoV-2, which are popularly used in the recent 1 year for development of the drugs. And definitely, some of the compounds from the plants will be identified. And in that process, the CADD has reduced the time of the drug discovery and development to almost 7 to 8 years. So one side, say you can see the target identification. So after target identification, we can do structure-based computer-aided drug designing or ligand-based computer-aided drug designing. There are two aspects. Structure-based, the target structure is there. Target means that receptor structure. Then we can do the ligand docking. Then we can do the molecular dynamics. Then we can do the optimization of the lead. Then we can identify the drug target candidate molecule. So this will be the research line. These are the two research line. Then another thing is, if we have the ligand structure with us, ligand-based computer-aided drug designing, so we can do the QSAR analysis, pharmacophore modeling, then we can do the ligand-based virtual screening and do the lead optimization, and finally we can identify the drug candidate. So selection of the small molecules for screening, as I have told, this is a very much prerequisite for a successful drug discovery program. Then selection of the drug targets from the literature. Then we can go ahead with the docking of the small molecule structure and the large molecule structure. So for both the cases, we need three-dimensional structure of the small molecules and three-dimensional structure of the large molecules. Now, many of you may have the question that we have identified the compound from GCMS, then how we can have the 3D structures. So while going through the compounds, while going through the databases of the identified compound, you will see some of the strings called SMILE strings. Okay, SMILE strings, canonical SMILES string, which can be used to reproduce the three-dimensional structure of a small molecule. So all public domain software is available from the web. You can identify the right software for you and you can use it. So this is one again example of bioinformatics and its application in drug discovery. So already I have mentioned these are some of the targets what we have used. So protein data bank, similar way that we did for Alzheimer's disease. So we have a lot of things, Indian Ayurveda, Unani, Siddha. So they have different recommendations for different diseases, including the SARS-CoV-2 or COVID-19 disease. So we can use those plants also for identification of the specific compound. Okay. So the use of those extract and the crude extract of the plant and the purified molecule, there is a vast difference. Because as I have already mentioned that the cup of tea what you have may not contain all the required medicinal plant in the right strength, medicinal compounds in the right strength, which is to be required to inhibit particular pathogen or inhibit a particular drug target more specifically. Okay, so we can have we can identify the exact molecule which will work. Then we can these are some tools what we can use. UCSF Chimera, CN3D, Rasmol, Pymol to review the three-dimensional structure of proteins, small molecules. Then we can select the base binding ligands. Okay, as I have mentioned about the Lipinski filter, an ADME-tox study we can do. So in silico, in the computer only, we can say that whether the compound will be toxic or it can be processed for drug in near future. So molecular dynamics is one of the key component of the binding analysis. Okay, we can see where the compounds will exactly bind in a protein. Okay, we can identify the active site, we can fit the molecule, whether it fits or not, that can be determined through a docking study. So as I have told, 2D to 3D, we can prepare model, then we can do the docking. So docking identifies the right conformation in the right place where it will best fit. Okay. So molecular dynamics will be discussed in our last presentation. Okay. So this is the example of a membrane protein. So anyway, but so big data analytics tools are being used. Uh, recently, because biological data is already a big data, so the platforms like NINE, okay, it can be very useful for big data analytics, typically in terms of drug AI-based development, based tools and techniques for right from the modeling of the protein structures to that of the identification of the right molecule for the right target. Then blockchain technology is a very new technology which is associated with the Bitcoin. So Bitcoin is a cryptocurrency that many of you may be familiar. So blockchain is a technology developed to maintain those cryptocurrencies. So that blockchain technology is used for identification of the for storing the information for medical purposes, for drug discovery purposes. So if you read some of the papers, you will definitely come to know it is not very complex. So just I'm giving a glimpse of it so that you can capitalize on this. So potential R&D use blockchain, deep learning technologies. Okay. So many of you may have one question that whether we'll sacrifice the plants and we collect the compounds for drugs. No, definitely not. So first, to identify, yes, we can use the small part of the root, shoot, or leaf or of the plant, but we can ask our chemist friends after identification of the ligand, say this compound is present in this plant, you please give a synthetic compound analogous to this. So he will definitely help you in designing the synthetic protocol for that particular compound, and he will produce the exactly same molecule which is available in the. That way we can produce in bulk. You no need to go every time go back to the nature to destroy the natural resources. That is what just general information that I want to pass. So now the big thing is the computational infrastructure, that what you need for starting a bioinformatics work. So you may be thinking that those who are not started yet, they may be thinking that it will require very high computational power, very high specs PCs, very hi-fi systems. Yes, it is absolutely not correct. So with a simple PC also you can start working. If you have a better specification, you can use more better software. Okay, 4 GB RAM is quite sufficient to work with most of the software except the molecular dynamics. So molecular dynamics, definitely it is very high specs of servers and workstation is required with graphics card and all that we will discuss in the last session. So these are some big servers and other setups, hi-fi bioinformatics setups, high performance and scalable computing resources, uninterrupted internet connectivity. So you can use already developed resources like IIT Delhi. They have supercomputing facility in the SCF Bio, that supercomputing facility for bioinformatics. You can request for a cloud use. You can remotely log in to their computers, to their supercomputer, and anybody can work, means by taking a space there. You can install your own software You can install, deinstall it. You can do the exercise. You can retrieve the result. Okay, that is not very difficult. So for software requirement, yes, we have for doing almost everything informatics based drug discovery, almost everything is available free of cost. Autodoc is a very if you go to the citations of the drug discovery software, so you will get the highest number of citation for Autodoc. Means most of the people, most of the researchers, they have used Autodoc. Then UCSF doc, these are free. Gold is commercial. Then for molecular dynamics simulation, we have the best of the molecular dynamics system, which is free, GROMACS, okay, NAMD. So there are some commercial also, definitely at the same time. But if you can do some kind of, if you can take little bit of pain, you can use the free software. Otherwise, the commercial software, they are very, very costly. So if you go to the drug discovery studio produced by earlier Acceleris group, it will cost you around 26-27 lakhs for a perpetual license. And Schrodinger, yes, that is one of the best that we also use sometime. So Schrodinger is very costly. That is yearly license basis. You can get for say 5-6 lakhs per year. You can spend if you have a project or on that, and you can take the advantage of their beautiful talking program, Glide, very nice molecular dynamics program that is Desmond, and very efficient homology modeling program called Prime. So that is choice is up to you. If you have a good project, good money with your institute, you can go for a commercial version also. There is no hard and fast. And another thing, if your institute or you do not have a high computational environment, always we can use the cloud infrastructure. So nowadays it is becoming little bit gradually it is safe, and if we learn, we have to know that how much space we require, how much work we will do. On the basis of that, you can customize your requirement in Amazon Web Services, popularly known as AWS or Microsoft Azure or Google Cloud. You can have meetings with them. You can fix your requirement. You can fix the prices. It's not very costly. But if you do not know how to use that blindly, so please don't go and open one account in AWS or Azure or any other cloud, commercial clouds. So you will be your credit card will be in trouble. Okay, trouble means it will be a very highly charged environment. But yes, if you know the features, if you know what to use, what not to, definitely these are one of the best, because you don't have to maintain a big server at your own place. AC rooms are not required, so no manpower. Lot of costs you can cut if you use the cloud infrastructure. And many of the software in renowned software, commercial software, they are coming with their web services also nowadays. So that's all from my part. So just I wanted to give a quick overview. Okay, it's just I'll tell overview. So definitely all of you are learned, all of you are educated. You can capitalize on the small things, and you can make a big difference to the country, to the entire human being, because we all have the potentiality that we believe that we have to learn. Internet is there. No book, nothing is required nowadays. All research papers, review articles are available. You please go through that. If you have the most foremost means most important prerequisite for CADD is your desire to learn. Okay, if you have the desire, everything is available, everything is free. You don't have to be a billionaire to start a startup. Also, even you can think of uh, getting a startup set up in any part, any part of the world. You can start your own work. There are a lot of companies which outsource different works like combinatorial library generation, docking, molecular dynamics, this thing, that thing. They outsource many part of the work which we can grab the opportunity if we do not sit, okay, idle. We can definitely have. Then there is a question, last question, what I can see, that NGS. Yes, NGS, the next generation sequencing, that is very analysis is very much popular. You can have NGS also in your basket, in your, say, entrepreneurship development basket, or you can have a very good career in, if you know how to do the NGS analysis properly. So the packages of bioconductor, R-based packages of bioconductor is very much popular for NGS analysis. Just you have the desire, you can learn most of the things. Okay, so I'll take up the questions, but the time is very limited. I can see some of the questions on the DRDOs, the 2Z drug. Okay, that is a glucose-based drug which is developed recently. Okay, the mechanism is not very much studied by me, so it is available in the. You can go to the PubMed. You can see their publication on that specific publication on that particular drug, and you can see the mechanism. It just, I want to know. I just know that it is a glucose-based drug which restricts the entry of the pathogen of the virus to the human cells. Okay, so I'll take up quickly some of the questions.